Okay, thanks everyone. So it's past, so I think we'll make a start. Welcome to this session on genome replication and transcription. We're going to have 4 talks before lunch, the first of which is from Jonathan Grimes from University of Oxford, who's going to be talking about influenza virus replication. John, the floor is yours.
Thank you very much. Thank you. So I'm going to be talking about some work we've been doing over the last few years on trying to understand how influenza replicates its genome. As a bit of context, flu is an RNA virus with a negative sense RNA genome. There are a number of RNA viruses that are important in terms of human health, and you can sort of view them in a number of ways. You can have viruses that have a segmented genome or a non-segmented genome. They can be sort of classified in terms of where they replicate and transcribe, and the bunyaviruses and, say, Ebola, rabies, they transcribe and replicate in the cytoplasm. Flu and Borna transcribe and replicate in the nuclease, and then you can sort of classify them on the mechanisms that they transcribe. And flu and the bunyaviruses use a technique called cap snatching, where part of the polymerase is able to capture the cap structures from cellular RNA and cleave off the cap structure. Whereas for the non-segmented, their polymerases have functions to enable them to synthesize cap. So this is just a rough overview of what flu looks like. It has a lipid bilayer in which you have the hemagglutinin and the neuraminidase proteins. You then have a matrix formed by M1, and this encapsulates the segmented RNA genome. Influenza A has an 8-segmented genome, as does B, and flu C and D have 7. And these are the things that I'm going to be talking about. A brief overview of what goes on during infection. The virus fuses, is endocytosed, and then by mechanisms that are not particularly well understood, these RMPs are translocate into the nucleus. And the polymerase has to do 2 related things. It has to transcribe viral messenger RNA, and in order to do that, the polymerase binds to the CTD of Pol2 and is there ready to capture cellular RNA. The viral messenger RNAs are then transported out of the nucleus, where new viral proteins are made. Some of these are then imported back into the nucleus, where the second function of the polymerase, which is to replicate a positive sense copy, and these new viral proteins form this new CRMP, and then there's a switch where the positive sense CRMPs are then copied back to the negative sense VRMPs. So the key block that we're interested in is the RMP, and this is essentially the viral RNA, which is encapsulated by the nuclear protein, and then the 5' and the 3' ends of the RNA are bound by the viral polymerase, which is a heterotrimer made up of PB1, which is the polymerase bit, PA that has the endonuclease domain that cleaves off the small capped messenger RNA, and PB2, which has a cap binding domain and several other domains, nuclear nuclear localization signals, and the like. So, to just reiterate, this polymerase has to transcribe, and how it does that is to bind to the CTT of pol 2. This messenger RNA produces viral proteins. These are imported in, and these are used during the replication process. So I'm now going to focus on what we've learned about that replication process. So a few years ago, Professor Barclay from Imperial College discovered that these proteins called AMP32 are key to influenza replication. The interesting part of this was that I didn't mention, but flu viruses are avian viruses, and you get specific adaptation mutations which allow them to become human. And a key mutation is this glutamate 627, which is on the PB2 subunit. In human polymerases, this is always either a K or an arginine. And what they were able to show in Imperial College is that if you take an avian polymerase or a human polymerase, that's able to replicate in chicken cells. However, when you take human cells and use a flu polymerase, with the human AMP32, you get replication. But with the chicken polymerase in the human cell, you don't get any replication. And we were able to show for the polymerase from flu C, because the structures I'll be showing you are primarily based on flu C. We were able to show that replication is the presence of human AMP32B or chicken is key for replication. If you don't have, so here we're using double knockout cells, and we're transfecting with human AMP32B or chicken, and we're able to recover replication. A little bit about human AMP32. It's found in the nucleus. People don't really know what it does. It has an LLR domain, and then from about 158 onwards, you've got this LCAR region, which is low complexity, highly acidic. So this is predicted to be completely disordered. The only difference between the human and the chicken is this 33 amino acid insertion, where, and you probably can't see this, but there are a number of a number of positive residues, arginines and lysines, in this 33 amino acid insertion, and this will be key to the adaptation. So we were able to solve the structure of the polymerase bound to AMP32, and we did this for both human and chicken. And I'll go into this in more detail, but you have a heterodimer of the trimeric polymerase. And the blue one is what we're calling the replicase, and the green one is what we're calling the encapsidase. So this is just a movie which highlights this. You've got the three subunits of the polymerase, PB1, PB2, and PA. And they've come together to form this stable heterotrimer. This is where the viral RNA binds. You have the 5' hook here, and then the 3' going into the active site. And we use synthetics In this structure, the cap binding domain is turned in, so it can't bind cap, and the PB2 and the NLS are up at the top. So this is what we are calling the replicase. For the second copy, it's rotated and comes in, and then you see these massive conformational changes in the domains in PB2. And then sitting between these two dimers is AMP32. This is a movie which highlights the important bits. So the product will be coming out here, and where it has to bind is in this little pore here. So this is a sort of replicating platform, which means that as soon as the 5' end of the product RNA comes out of the replicating polymerase, it's immediately bound by the encapsulating polymerase. So you don't have free RNA. Now, in terms of the adaptation and the adaptive mutations, so when we contour down our maps from our cryo-EM, we can see at low resolution a tube of density running between the two 627 domains that have come together. And right where this insertion is, you get the key adaptive mutation K in the flu C. It's 649, but structurally it's equivalent to K627. So it's these two adaptive mutations which somehow allows this highly negative bit of the human to be able to bind. What we also know is that this bit of LCAR is key for viral production. So when we truncate the LCAR, we lose RNA production. And we see this very clearly when we have longer pieces of RNA. Not only is it key for production of viral RNA, but when we truncate AMP32, we can see that, and this is using a pull-down assay, we lose an interaction, a direct interaction with the viral nuclear protein. So there's some role for LCAR in binding to the nuclear protein. And what we can see is that this bit of LCAR is actually key. Actually binds to the RNA binding groove on the nuclear protein. So there's a positive groove on the nuclear protein of flu, and called the G1 and the G2 groove. What we're able to show is that in the absence of RNA, we're able to pull down nuclear protein via AMP32. But when we add in RNA, we lose this interaction. When we use our mutant MP, which can't bind RNA, we also see that we strongly lose this sort of direct interaction to the nuclear protein. So we're able to put together now a model of what's happening during viral replication in the nucleus. Newly synthesized polymerase comes into the nucleus and binds to the replicating polymerase on the RMP, and this is stabilized by AMP32. The newly synthesized product is the 5' end is immediately bound in the encapsulating polymerase, and nucleoprotein binds to the RNA, protecting it. And key to this is this this low complexity acidic tail, which is able to increase the local concentration of nucleoprotein around the RMP and pass this RNA-free nucleoprotein onto the encapsulating RNA. And this is sort of analogous to what's seen in the non-segmented viruses, where you have the P protein, which is a sort of molecular chaperone. It binds to the polymerase, ends of it bind to the RNA-bound nucleoprotein, but the N-terminus of P is able to bind free, RNA-free nucleoprotein, which is thought to then be able to increase the local concentration. So I won't go through the summary particularly, but just to sort of, this little movie summarizes, I think, where we've got to. So this is our model of an RMP. The viral RNA is in red. The encapsulating polymerase comes in. AMP32 stabilizes this, and then you've got this flexible acidic LCAR, which is able to capture RNA-free nucleoprotein, and then, as the RNA is produced, this RNA-free nucleoprotein is then able to encapsulate the newly formed RMP. So this work would not have been possible without a fantastic collaboration with Erwin Fodor at the London School of Pathology. And we have a really great sort of group that works extremely collaboratively together. And this would also wouldn't have been possible without funding from the MRC and the Wellcome Trust. Thank you.
Fantastic talk, John. Yeah, and thanks for staying well within time. So we do have time for a few questions at the back. Yeah.
So why do you think the here AMP32 is deliver the nuclear protein to the RNA? Because like MP might be there are plenty. It can diffuse to bind to RNA. What if the role is actually taking the MP out of the RNA so that it can go into the polymerase or something like that?
Well, the viral RNA is always bound to the nucleoprotein. It has to be pulled off when the RNA is dragged through. But what you don't want is the encapsulating polymerase to have RNA hanging off it. So, I mean, it's just a model, but it's plausible that you want to increase the local concentration of RNA-free nucleoprotein around the replicating complex, and the nucleoprotein will bind RNA just like that. So you need RNA-free nucleoprotein in the vicinity to be able to encapsulate the newly formed RNA. That's the model, anyway.
Question as well at the back.
Is in the avian system, the AN insert, is it in any way more flexible or better to deliver to the RNA to make, because it's so much more pathogenic with a wider organ host range?
Yeah, I don't know. I mean, there are only sort of 3 or 4 arginines and lysines in that insert. So, you know, it's a bit hand-wavy. I mean, it is a bit hand-wavy, but it's clear that these adaptive mutations occur right in this 6 to 7 domain, which are brought together in this replicating platform. So, yeah.
Maybe time for one more quick question, if anyone has one. If not, maybe I'll ask a quick question then. So this is broadly applicable to other influenza viruses, is AMP32, right?
Yes.
So what does this kind of AMP32 look like in potential intermediate species? So, for example, you see a lot of influenza now spilling over into mink farms and things like this.
I mean, AMP32 is found in all, in all. In fact, it's found in fish. So they're now discovering influenza viruses that infect fish, and AMP32 is found in fish. So I mean, it's a very conserved protein that's found, in all nuclear.
Yeah. All right. Thanks. Okay, so in the interest of time, so let's thank John again. Yeah. And our next speaker is going to be Reza Khayat, who's going to be talking to us about the structure of the Circoviridae replicase. The floor is yours. Clicker? Oh, John, did you steal the clicker?
Oh, here it is. All right, let's see if my luck is better. Okay. All right, thank you to all the organizers for allowing me to speak, and I guess I'll begin. So this is a project we started a little bit before COVID started, and we made good progress, and then there was some slowdown, but we picked it back up. And there's a lot more left to be done with this project than what I'm talking about. So what are Circoviridae? Circoviridae is a family of viruses that have two genuses, and the name comes after the single-stranded DNA circular virus that they have. So Circo stands for circle. The two genus are Circovirus and Cyclovirus. Both are ubiquitously present in animals. Circovirus is associated with a number of diseases in various animals. PCV in particular has the ability to jump from one animal to another. Cycloviruses tend to be more present in humans. What disease they cause, we don't know anything of, but we know that they're just running around within us. As I said, they have circular single-stranded DNA genomes. Both encode for two major open reading frames, a replicase, which is the topic of today's talk, and a capsid protein. Now, interestingly enough, despite the fact that the two genes are transcribed in different directions in the two genomes, they share more than 40% sequence identity. So they are homologous to one another. They both have high mutation rates and recombination rates, and that's believed to be due to the replicase. How it does that, we don't know. And one thing I want to mention before I go a little bit forward is that when the replicase was first identified, it was identified because it was required for replication. But as you'll see later on, it's not really a replicase, it's more of a helicase. And it requires the DNA polymerase of the cell for replication to occur. And the last interesting fact that I really like, these are the smallest or the simplest autonomously replicating viruses. So you have genomes that are smaller, for example, AAV, but adeno-associated virus requires another virus for infection. These ones infect by themselves, they cause disease by themselves. So it's a beautiful system for understanding what is the minimum amount of biochemical information you need to generate an infectious virus. So PCV, the first genotype, was identified back in the early 80s. It was found to be chronically infecting porcine kidney 15 cells. So it was named porcine circovirus. The capsids shown here above are about 17 nanometers in size. They ran SDS page on it, found that the major capsid protein was 36 kilodalton. That's the only capsid protein. And then they used some palladium, platinum, metal shadowing and compared the size of the genome from PCV to 5x174, which we know is a single-stranded circular DNA. And they identified that maybe the genome is somewhere around 0.6 megadaltons or around 1,700 nucleotides. So right off the bat, they got a good amount of information from the virus. So what we're interested in is we're interested in understanding the entire life cycle at atomic resolution. This involves everything from the capsid structure, cellular attachment, entry, trafficking to the nucleus, delivery of the genome into the nucleus because it's a single-stranded DNA virus, so it requires the machinery of the nucleus, assembly, aggression, and so forth. And we've made reasonable progress on a number of these steps, but today's talk will be on genome replication. So how does genome replication occur? So it occurs through rolling circle replication, which was which was coined for describing how phi X174 replicates. So we think this is, again, a very simple system to start understanding how rolling circle replication works. So it's clearly nowhere near as well characterized as phi X174, but it's a slightly different system, so we're pursuing this one. And before we started working on it, functional and structural and prediction bioinformatics approaches suggested that it had two domains. ED, which stands for endonuclease domain, and AD, which stands for ATPase domain. The endonuclease domain is well characterized. There's an NMR structure, there's a crystal structure with the substrate attached to it. The ATPase domain was predicted, but, and this is where we start bringing in we contribute structural information to it. So how does RCR occur? Well, it's a single-stranded DNA virus. It makes it into the nucleus, and somehow you get double-stranded DNA. Now, how does this happen? We really don't know, but there's one publication out there that says there's a small piece of DNA attached to it, a complementary strand of DNA that acts as the primer for making the second strand of DNA. And it's at this point, when you have a double-stranded DNA, that the replicase becomes involved in replication. So what does the replicase do? It binds to a particular sequence called the origin of replication. It remodels the DNA to generate a cruciform, and then the endonuclease domain nicks the stem loop, or the loop of the stem loop. It becomes covalently attached by it, and then it starts separating the two strands of DNA. Now, at this point, it's not known how many polypeptides are involved, whether it's one molecule or one oligomer that's doing the job, or if it's multiple molecules doing the job. And this is what we're hoping to understand. And this proceeds until you get a full round plus some additional nucleotides rounds of replication, and then that endonuclease domain picks up the free end and ligates it to the end that's attached to it and releases the genome that's ready to be packaged. Okay? So what's being replicated is the material that's actually going into the virus. And during this entire process, the capsid protein is likely to be binding to the single-stranded DNA genome, because this is a single-stranded binding protein, and it's helping probably package it into the capsid, although that's just one possibility of what may be going on. And then the process is repeated again and again until you get aggression occurring and so forth. Okay, so you have an endonuclease domain and an ATPase domain. So we set out to start expressing, characterizing this. So we got two ways of doing this, either cryo-EM or X-ray crystallography. We were not able to express enough of it for crystallography. It's a 314 amino acid polypeptide chain. We ran a size exclusion chromatography for the polishing step, and it suggested that the molecule was a little bit bigger than a hexamer. So we decided, well, it's big enough, let's put it on a grid, and let's see if we can see this stuff. So we put it on a grid, and initially we had some problems with preferred orientation, and we still have those problems. But what you can do is you can simply tilt the stage and deal with that problem and get multiple orientations. So we did that, and in the 2D class averages, we saw that we do have a hexamer. What we also saw was we saw side orientation, and we saw that there's this fuzzy density that may be contributed to some dynamic domain that is just not ordered and we can't visualize. So this is the beauty with EM, is you can, at every step, you can identify something, you can learn something about what you're studying. So Took this, just pushed it forward. We got a 3D reconstruction to somewhere around 3.2 angstrom resolution. Now the resolution varies. You have parts of it that are about 2.5 angstrom resolution. But we have something that looks like to have 2 domains, and it's a little bit smaller than the anticipated size. So what's going on, and seeing this fuzzy density back here, we decided let's try some 3D variance analysis, try some classification, see if we can get anything out of it. So none of the traditional classification methods, whether it's focused classification or anything like that, worked out, and only 3D variance from CryoSpark worked. In fact, we couldn't solve the structure with Relion. I don't know why. But we got a couple of different densities up here. Right, and that's telling us that there probably is a domain, and that domain is flopping around. So looks like we have a 3-tiered or 3-domain molecule that forms a hexamer. As you can see right off the bat, is that as I rotate this, is that it's not a C6 hexamer. It's a C1, it's asymmetric. There's something bound to the center of it. This isn't something we added, it's something that was bound when we purified it from the crystals, from the E. coli, and there's a seam between 2 of the subunits, okay? And you also see that the subunits seem to go around in a staircase arrangement. So take this, here's a couple of additional orientations, and what we're looking at is we're looking down that pseudo-axis of symmetry, or axis of asymmetry, and you can see there's that gray material in the center. There is the seam I was talking about. Here's the backside showing that there is this arrangement of this domain going around and spiraling down. And here you're looking at the top, at the bottom again, or you're again looking down the other side of this asymmetric axis. So what you can do is, obviously this was done after the tracing was done, or else we didn't know what the polypeptide chain is. But you can separate the 6 out and put the ODs on the same level, and you can see, or this domain on the same level, and you can see that you have 2 domains. One we call the oligomerization domain that's responsible for oligomerizing the hexamer. And you have this ATPase domain that seems to be moving up as you go around the circle counterclockwise in this direction or clockwise in this direction. So this is giving that spiral staircase arrangement that I was discussing earlier. So what does the structure look like? Well, here's the structure of the OD and the AD. We can trace the polypeptide chain from amino acid 119 all the way to amino acid 301. So it's a 314 amino acids. We can't see the first 118, which is not that terribly problematic because there are crystal structures of it out there, so we can make the pseudo model of it. And we also don't see the last 13 or 14 amino acids. These are disordered. We have a bunch of additional structures that we see this, the rest of the C-terminus on, and that creates an interesting story, but I won't talk about that for this talk. So the OD, if you do a Dolly search, it's homologous to homeodomains. These are domains that proteins use for interacting with nucleic acid. We think the nucleic acid goes through the center of rep, so it's doing the same function here. And the AD is the canonical fold, A alpha beta alpha fold that you see for AAA plus ATPases. What are these? These are ATPases associated with various cellular activities. These are essentially, it's what PJ was talking about, and these are essentially ring-like molecules that use NTP binding and hydrolysis to pull material through the center of the pore. What is the material? It could be nucleic acid, as in this situation, DNA, protein. It's used for digesting protein. Proteasome is one of these. It's used for pulling proteins apart. So it's used for pulling things through it. That's just a very generic description of what AAA plus proteins are. So again, here is that's when we assign this density to be the ED to the endonucleus domain. So this domain is highly dynamic. And is that dynamic nature important for its helicase activity? This is one of the things we're pursuing at the moment. Okay, so if we take a closer look at the structure, here again we're looking down that axis of symmetry. And in our structure we see, we're with AAA TPases, the ATP binding site is between two subunits. So you have 6 ATP binding sites. In those 6, we see 4 molecules of ADP bound, and we see 2 empty sites. And I'll talk about what this means in slides to come. What other things we see is that the single-stranded DNA in the center, and we see that there's 2 loops from rep that interacts with with the phosphates and then the phosphodiester backbone of the DNA. Of the 6 subunits, all the subunits going from the top of the staircase to the bottom, these 5 are interacting with the DNA. The last subunit is loosely associated with the DNA. Now, this is important for the mechanism that I'm describing. I'm going to describe for how it does its job. And here, we're looking at that spiral staircase going around. And remember, each one of these different colors is associated with a different polypeptide chain. Okay, so here is that mechanism that I was talking about. So the field at this point, I don't want to use the word split, but there are 2 major mechanisms being proposed. There's a mechanism that PJ discussed, which some would call an all or nothing, where it goes from this washer look to a cracked washer to a flat washer, or the inchworm model, which is the name I like a little bit more because it's cute. And then there's this hand over hand model, in which you have one hand at the bottom, you have 6 hands, and then one hand, the hand at the bottom goes to the top, grabs, and then all the other hands pull down. And there's an animation here that's showing that. Okay, so this is this subunit goes through each one of these different conformations to move the helicase 1 nucleotide at a time, going from the 3' end to the 5' end. So that's one thing I did not talk about, is that there's directionality in this. It's been shown biochemically, and now we have a structure showing which direction it moves. Okay, so the idea is that, again, this has not really been shown. It's just been proposed. You have ATP here. You have 2 ADPs, 2 empty sites. The incoming ATP goes between the seams. The subunit at the bottom goes to the top, and everything else goes down, and this continues until you travel across. So this is a dynamic process. We need to use dynamics to understand it. So I decided to, cryo-EM uses really good, can give you millisecond dynamics, but not really anything faster. And I think I'm really interested in the chemical question. So we decided to turn into using molecular dynamics. So we generated a full rep. The full model was used using a template of a homolog to it. And we're comparing rep with ADP bound to single-stranded DNA, rep bound to single-stranded DNA, and just rep by itself. So what is the first question? Well, if MD is going to work, we need a control. The control is, does rep bound to ADP plus ssDNA generate the structure that we have? Is it comparable, or is it completely different? So as you can see, after 700 nanoseconds, you still maintain that staircase arrangement. So that says that, yeah, the control is working, so this is probably a way to go forward. We take the nucleotide out, staircase is maintained. We take the DNA out, and the staircase collapses to that washer structure. So what's driving the staircase conformation is not the nucleotide, it's the single-stranded DNA. And that's because of the interaction between the subunits with the DNA. So one way to show this more simpler is to draw a plane between those lysines that I showed earlier, and just simply run an animation of those planes. And you can see that for these 2, the plane remains tilted, but for the apole, where both the nucleotide and the DNA is lacking, it's becoming flat, and at the very end of the 700 nanoseconds, it adopts the flat conformation. So this simulation, despite my postdoc's suggestion, actually, in my opinion, seems to have worked, and it's given us a wealth of biochemical experiments to try to, that I would not have been able to predict just by looking at the structure, to try and understand, see how the rep works better. So and here you can plot these angles by comparing the angle between this plane and this plane, where you start and where you end. And you can see that for ADP and ssDNA bound, it's fairly stable. When you take ADP out, it's showing a little bit more variance, and that may indicate that ADP is actually being used to stabilize the interfaces. And if you take the DNA out, then it just collapses to something that's more flat-like. So with that, I draw my talk to a conclusion. This is essentially all of the collaborators we've had for PCV2, because then, again, we're looking at many different things. All the heavy lifting for this project, for this particular project, has been done in this lab. And I'd like to thank the NIH for funding and NSF. We have an NSF REU program that a number of undergrads have worked on this. And of course, all the data has been collected at the NYSPC, the Simmons Electron Microscopy Center, and City College for initial funding. And if you have any questions, I'd be happy to answer.
Great talk.
Thank you.
First question at the front.
Yeah, great talk.
Thank you.
Do you know anything about the affinity for DNA depending on the nucleotide state? Is it higher with ATP bound versus ADP bound, for instance?
I don't have any idea I do not have. These are experiments that I have not tried. And again, like I said, the DNA that's attached to it came purified with the sample. So I'd have to strip that out and then do these experiments.
Yes, because you have ATP and it's the staircase, you must have a reasonable affinity for DNA. You know what I mean?
Yeah. So one of the other questions is that how do you get a hexameric ring around a single-stranded DNA, right? So this is a big problem. It's not a big problem, but this is a problem that people have been studying in E. coli replication. There's DNA B that has the same problem. And there's two mechanisms. There's a ring breaker and there's a ring maker. So in this system, because it's not endogenous to E. coli, it's foreign, I think this is a ring maker situation, and how it's interacting with that DNA, whether there's a particular sequence in the E. coli that allows that ring to be made, these are questions that need to be answered. But it's probably a ring maker rather than a breaker.
Yeah, super cool.
Thank you.
Cool stuff. So you mentioned that there's some specific sequence that is detected at the start of the replication.
Yes.
Is it this bit of the protein or the other domain that you didn't see but is crystallized that detects this specific sequence? And can you, from your structure, see how does that work?
So excellent question. This is one of the questions we want to answer. I've looked at our structure left and right. There's nothing I can predict. We've been able to bind it to the origin of replication sometimes it doesn't bind. I think it's just something we have to work out. But this is the big question. I mean, everybody in the field is trying to get structure of a helicase bound to the origin of replication, to anything, to get an idea of how it does this remodeling. Because in all the situations, the helicase has some role in remodeling the origin of replication. Yeah.
One more quick question.
Okay, so in Phi-X, there's a pass site, and there's actually a host factor that comes in pre-A and then helps recruit other factors that start this. Do you think there's a host factor that might recognize that sequence and then bring in the helicase? Or?
There could be, except there's a particular author that's published some very interesting stories, and they've shown that you can get rolling circle replication like replication occurring in E. coli, where this host factor is lacking. So I've never been able to repeat these experiments, but maybe I just don't have, my hands are not good enough. But according to that paper, it seems that there's no host factors don't seem to be necessary for this to occur.
Great talk, thank you.
Thank you.
Thank Reza again. And our next speaker is going to be Eric Dykeman from the University of York, who's going to be talking about modeling translational control of gene expression in viruses. The floor is yours.
All right. Thanks to the organizers for allowing me to present some of my work today. We've heard a couple of nice talks about the replicase and replication of the RNA genome. So here I'm going to switch gears and actually talk about the translational apparatus and how that's regulated in the cell. And one of the model systems that I'm interested in is phage MS2. And I think it's a very simple system that gives some insights into how single-stranded RNA viruses can control their replication via RNA secondary structure. So this is my cheap sort of simple drawing here of the replication cycle for MS2. So you have some sort of so the phage attaches to the pilus via the maturation protein, which then allows the RNA, positive sense RNA, to get inside. Once inside, pretty much the ribosomes that are in the cytosol immediately pick up the RNA and start translating it as a messenger RNA. The two main proteins that are involved in phage MS2 are really coat protein and RNA-dependent RNA polymerase. And so what's interesting is because single-stranded RNA viruses have no DNA stage to their infection cycle, all of the regulation and control of the quantities of viral proteins and the replication process all has to be controlled via the secondary structure of the messenger RNA. And so basically, coat protein, RNA polymerase is an early gene, whereas coat protein is sort of a late gene. And so eventually what happens is that there's a feedback mechanism in which coat protein binds to the replicase or start codon, and that blocks further synthesis of the polymerase. And then that sort of switches everything into a packaging stage. And so basically, you need 1 copy of maturation protein per RNA. About 5% of coat protein is actually equivalent to lysis concentration, and you only need RNA-dependent polymerase produced early in the infection cycle. So the question is, how is all this controlled temporally? And so a lot of this was worked out by Van Duyen and his 3 PhD students in the 90s, where they looked at the secondary structure of the phage MS2 genome in quite a lot of detail using enzymatic probing and phylogenetic analysis. And what emerged from that experimental work was essentially they found that there's 4 genes in MS2, and essentially all 3 of the genes are hidden from the ribosome. And so only one of them is accessible to the ribosome, and that's the coat gene. And so this is sort of a cartoon diagram of the coat lysis, so the 3' end of the genome. And if we blow up this section here and sort of stretch it out, then you can see the coat start, which is hidden in this hairpin here, and then which is the RNA ribosomal binding site. And so basically in prokaryotes, the ribosome wants to bind predominantly single-structured areas of the RNA. And so this provides a sort of area in which the ribosomal S protein S1 will bind to this area and then initiate on the start codon for the coat gene. But if we blow up the replicase start site, what you see is that the structure of the secondary structure of the viral RNA actually sequesters the start codon in this cloverleaf structure. And so the ribosome has no access to that. And so this gives control over gene expression via what's called translational coupling and repression. And so that's sort of demonstrated here in this cartoon. So basically you get the ribosome initiates on the coat protein start site and then starts translating. And then you get co-translational folding of the 5' end of the ribosome and melting of the 3' end. But eventually it encounters this Min-Ju sequence. And that opens up through melting of the RNA structure another binding site, which is predominantly single-stranded, which then allows the ribosome to initiate on the replicase gene and make replicase. But later in the infection cycle, you get coat protein will actually bind this TR hairpin, and then that prevents the ribosome from melting this structure and accessing the start codon. So eventually it shuts off the synthesis. Okay, so basically to understand the viral life cycle, in phage MS2 at least, we need to understand the interplay of RNA secondary structure and gene expression. And so I started this sort of from looking at basically the ribosome. So how does the ribosome interact with the RNA, and how does it move over the mRNA template? And so the first goal was to build sort of a holistic model that looked at ribosome movement on messenger RNA based on the knowledge of the kinetic steps in their rates. And then the second goal was, once I created this sort of stochastic model, was to somehow incorporate the RNA secondary structure and its interaction with proteins and ribosomes. So the ribosome kinetics in vivo is quite complicated. So this is a sort of schematic diagram of all the processes. But it can be basically broken down into 3 stages. So you have the initiation, elongation, and termination and recycling steps. So in this system, you've got several cofactors. So you've got initiation factors, IF1, IF2, IF3. You have the elongation factors, EFG, EFTU, and EFTS. And then you also have tRNAs. And then you've got the recycling factors, so RF1, RF2, RF3, and ribosome recycling factor, RRF. So that kind of gives you the details of the prokaryotic system. And so all of this is highly concentration dependent. There's roughly in prokaryotes, so in E. coli, which is doubling, say, every 30 minutes or so, you'll roughly have 60,000 ribosomes and about 5,000 messenger RNAs in the cell. So that's kind of the background. And the goal of this was to incorporate as much of the kinetic reactions that are known. So many groups, Ehrenberg and Rodnina and several other groups have worked out basically all of the kinetic steps with quite detailed biochemistry of all of the individual steps that take place and a lot of the kinetic rates. And so basically using that data, you can construct a stochastic model which looks at the ribosome moving through each of these individual steps in a reversible manner. And then with the addition of putting in the cofactors, so EFG, EFTU, et cetera, you can build sort of a competition between these factors for access to the ribosome. So for instance, here, if you have the elongation state, you can always bind recycling RF1, so a release factor, or you can continue translating by binding a ternary complex, and so go through the process. But this switch here, of course, is rate dependent. It will be very quick if you've got a stop codon or et cetera. And so basically, this was my first project to build all of this model, and so that was done. And basically through sort of very specialized computational techniques, you can simulate translation on 60,000 ribosomes on 5,000 messenger RNAs in roughly about 24 hours on a single CPU. So that allows you to then to sort of look at this in a reasonable computational time frame. So then the next step was to somehow incorporate RNA secondary structure into the folding kinetics into this. So basically, the big idea is that you've got the transcriptome of the cell. And so you've got 5,000 host mRNAs that are being translated by 60,000 ribosomes in the host cell. Then all of a sudden comes along a virus, and it inserts one of its messenger RNAs. And so then the question is, well, what happens during the infection cycle, and how does that impact the host ribosome and the host metabolism? So that's kind of a big, broad question that slowly, over time, that I'd like to work towards. But the first step there is to somehow incorporate the actual mRNA structure. And so that came along, which was the idea, which was to have a background of cellular RNAs, which just basically are just single genes. But that gives you the background of what's happening on each of those genes. And then you put in a single dynamic RNA, which you can then simulate the co-translational folding as the ribosome moves over it, and its response to proteins. So this is kind of the little bit of math that's in here. So the idea is that you actually have the full-length mRNA structure in the computational code. And then you can store that in sort of a specialized tree. And then using this, that allows the computations to be done very quickly. And so here's an example where you have the ribosomal footprint here in yellow, and then the structure of MS2 messenger RNA. And then here you've got a series of hairpins, and then this is in the process of refolding. So this hairpin in blue, there's actually a more energetically favorable state to be in, which is this one. And so you get a transition in which that hairpin will refold. And so basically, as this ribosome moves, you get a melting of the 3' end and a refolding of the 5' end. That's all taking into account the nearest neighbor models that you get for Turner et al. Okay, so the ribosome, you can also put in basically the ribosomal protein and protein binding reactions. So basically in prokaryotic systems, you have what's called a standby site. And so you can have a single predominantly single-stranded region of the messenger RNA in which the ribosome comes by and sits via binding of the S1 protein and waits for that hairpin that I showed you earlier for the MS2 coprotein to melt. And so you can create a straightforward model of the coprotein expression, which follows this curve. And this curve then depends on the melting time for that hairpin. So basically, you're looking at how much expression do you get for how long it depends on how long it takes for that small hairpin to melt. And so if you plot that melting time, you get sort of this sigmoidal curve. And so it's at this point here, the melting time is basically so short enough in which the gene is fully saturated. Okay, and then you can, this is just a little bit of a side that since we know so much about MS2 and its binding to binding of coprotein to RNA, you can put in a straightforward kinetic model for the binding of coprotein to the TR hairpin, which is sequence dependent. Okay, so putting all that in, D. Smit and Van Duyn in 1996 were looking at expression levels of the coprotein gene. So this is relative expression relative to wild type. So you have the wild type hairpin here, and if you make mutations to the coprotein hairpin stem loop here, you get different expression. So basically, if you make that, so on the previous slide, so if you make the melting time of that hairpin longer, okay, you get less expression. If you make it shorter, you get more expression. Okay, but only up to a certain point. And what they showed is that if you make this hairpin stronger, you decrease expression. And so you can plot that against the model, and this is the experimental plot of the model. And then if you fit that then to the translational model and my formula here, then you end up seeing that, so the red is the experimental measurements and the model prediction. So it fits quite nicely to that. Okay, so that was the concept there. And so then now this is the actual simulation of basically, if I go back a couple of slides, oh, maybe not that far, okay. So basically the entirety of that MS gene that the coat and the RNA polymerase. And so here you can see what happens is that coprotein is being synthesized, and then early on in the first 5 minutes or so of replication, the RNA polymerase is sort of synthesizing at this rate that's given by the dashed line. But eventually, coprotein starts increasing in concentration, and it starts binding that TR hairpin. And so you can see the replicase starts to get shut off. And so then this is a measurement, or a theoretical measurement, of how the replicase and coprotein concentrations in the cell would be temporally achieved by the virus as the infection cycle goes on. So you get that shut off. But what's interesting is if you play with this coprotein hairpin unfolding time, you can see that there's actually, there's more than just a coupling to the coprotein. So this is coprotein expression. You also see that polymerase expression changes dependent on that coprotein hairpin. So that it's more than just coupling. There's also a downstream dependence, and there's a optimal sort of thing there. Okay, so what I wanted to end it on with is sort of my forward. Okay, I just have just enough time into a little bit of experimentation. So I'm principally a modeler, but I'm also interested in dipping my toe in the experimental pool. So this is just a very straightforward, simple experiment, I think. So the question is, can you create a general messenger RNA that's designed to control gene expression just like an MS2? And so just to do something simple, hopefully, what I thought is that I'd try and replace RNA-dependent polymerase with a luciferase. but make the luciferase under the control of MS2 coat protein. So then if we cut out the gene that's associated with the RNA-dependent polymerase and replace it with a nanoluciferase, then you get a new structure here. And then if I just take out basically the coat gene plus the nanoluciferase, I can then I put that in on a PET vector under the control of a T7 promoter. And it's a bit, the background's a bit dark here, but hopefully you can see that it expresses and it fluoresces at 460 nanometers. But the size of this is just 1,000 nucleotides, about 1.1 kb. So the messenger RNA just has the two genes, which should be coupled, hopefully. So interestingly, if you put this into the model, what you see is that the model suggests that there's a problem, and that it predicts that when the ribosome moves over the RNA binding, ribosomal binding site for the nanoluciferase gene, there's two potential co-translational folds. So one, it represents the original structure, whereas the second here results in an off-pathway structure, which further sequesters the start site for the nanoluciferase gene. And so it predicts that you're going to get sort of low expression because of that. So the question is, can this be fixed? And so basically the idea is then to recode the messenger RNA to fix it so that we don't have that problem as with the co-translational folding, and so that basically it always refolds into the native ribosomal binding site. And so then, I can fill in the gaps on this if you ask me during questions. So basically, if you do the experimental measurements, so here is the original sequence which was uncoded, but then recoding it and testing it with my program, basically you can increase the expression of the nanoluciferase 7 to 10-fold. Okay. And so then if you compare with the model, so the model predicts that because of this recoding, there should be a 5-fold increase in protein expression before it's shut off. And then experiment says that there's about 7-fold increase. So roughly in line. And so then finally, you can do translational coupling and repression. And so basically, it's just going to show that my nanoluciferase is now under translational repression and translational coupling. So you can introduce this mutant, which is N55D, to the coat protein, and that doesn't bind RNA. And so just as you'd expect, because there's no shut-off of that gene, so when you have the N55D mutant, you get 30-fold increase in nanoluciferase compared to the recoded version. And then you can also put a stop code on prior to that Minju sequence to prevent that from opening up. And then you see that you get a small amount of expression, so it's a little bit leaky. But if that was fully open constantly to the ribosome, then you should see expression at the same level as the N55D because the coat protein is missing. Okay, so just wanted to end and give a massive thanks to Fred, who is very gracious, and all of his lab members as well, for allowing me to do my little bits of experiments on their lab benches, and to Alex Borodovka in Cambridge, who also gave me some lab space, and the EPSRC for funding my little adventure.
Thanks, Eric. We do have time for a couple of questions. Yeah, Pranav?
Great stuff, Eric. I have a very naive question. So you now have a phenomenon, you have a readout, say your luciferase fluorescence. Can your model predict when that event would happen upon changing the amount of input virus, for instance? So if you have an MOI of 1, the event happens 20 minutes post-infection. If you have MOI 10, does it now happen at 2 minutes post-infection? Is there a relationship, and is it?
Yeah, so actually that's my next thing. That's like a side experiment that I had thought of, which is if you deliver this just as with MS2 into the cell somehow, and then you could look at MOI. And because then you would have an indication of how many viruses get into the cell during an infection. And because it's then proportional to the fluorescence, hopefully you get a histogram distribution where you see, a lot of the times it's 2, or sometimes it's 1, or whatever the distribution is.
And also the time dependence or the temporal correlation between the input.
Yeah, exactly, yeah. The only difference with this is I'm not sure if you can get the substrate into the cell. I've been sort of researching that, if you can get it in. Otherwise, you might have to switch to GFP or something like that.
Okay, yeah, one quick question.
So very nice talk. Maybe I misunderstood, but is a possible goal to use this technology or idea to improve protein expression, protein recombinant expression?
Yeah, so I think it's twofold. So one is, if you've got a protein that's, I've heard sometimes experimenters say, my protein's not expressing very well. So my hypothesis, if it's in a bacterial system, would be there's something wrong with the ribosomal binding site. It's not presenting the correct structure to efficiently express. And so then you could use this tool to sort of try and adjust the structure of the mRNA through synonymous coding to get you a better structure that might express better. Then the second reason would be to, a gene circuit. So if you wanted to deliver messenger RNA somehow into the cell for expression, you may not want to have it just keep pumping out that protein forever, okay? So then the question is, well, how can you control that so that you get maybe a cutoff? And this is one example.
Okay, so can we thank Eric again? And the last speaker of this session will be Paulo Tavares, and the title of his talk is Cellular Compartmentalization of SPP1 Genome Replication and Viral Particle Assembly. The floor is yours.
Thank you. Okay, so first of all, I would like to thank the organizers to put together the PVA. This is a great place, the convenient weather for being the conference room, and last but not least, I thank them for giving me the opportunity to present our work. So, in fact, this is the final result in case you want to go for lunch, but maybe I will put things into context before. So this is the way to go. So, I mean, we have been working on SPP1 since a long time, and we had a lot of in vitro systems that were established. I mean, we could just make the ejection from the phage with purified ectomen of the receptor. The work done on DNA replication by the Juan Alonso Lev and Silvia Ayora, they really use the purified components to really reconstitute in vitro all the initiation events of replication. We made a lot of experiments also in viral assembly. So we have, in fact, a pretty good idea of what's going on in vitro. But probably, I think, in the middle age crisis period, I decided to think what's going on more when we try to put this in a more cellular context. I mean, what are the constraints and the advantages of having it? So we start to put setting up a series of tools that allowed us, in fact, to look to all this process in the cell, and that's the interest of this talk, which would be rather descriptive of what's going on. I want just to let you know about Audrey Labarre. That was really the driving force behind all this setup. And the first thing that we saw when we do a transcriptome of the SPP1 genome, it's really fascinating how you can see that the highly transcribed genes from pretty early during infection are those exactly that are needed for assembly of the virion itself. So this is the most required bricks to enduring the phage infection are those actually that are made to build the capsid, the tail, some genes here also for lysis, And there is always an outlier, like a small RNA, but whatever. So this tells us that we want now to know where these processes are going on. And the first experiment we made was to put some numbers in the infection process. So this is a qPCR experiment in which we quantified the number of genomes that are synthesized in the cell as the infection is made in rather artificial conditions, 37 degrees LB, everything goes fast, replicates very efficiently. This starts the replication around 10 minutes, grows exponential till lysis. DNA packaging follows, I mean, this like as you'd expect from the books. And we can also see that, so how this goes on. So this summarizes the situation, and we can then follow the formation of particles with DNA by making DNA's protection before the qPCR quantification. And finally, we correlate these to the number of infectious particles, and we come to numbers like 60% of the total sp2 DNA is protected inside various at least lysis time. So when we look then to what's going on in terms of DNA synthesis, we can see this is the major biosynthetic challenge which the cell is exposed to when we synthesize around 13.4 megabase pairs of DNA within something like 15 minutes. So this is much higher amount of DNA proportionally to what we have in the cell, and sp1 does not destroy the bacterial DNA, than the amount of viral proteins that we synthesize relative to the total number of proteins in a cell. So the major real challenge for the cell is that that during sp1 infection, the total DNA doubles within a short period. And this also comes to the point that the cell volume we measure that does not change during infection. So this means you just raise a bit osmotic pressure inside the cytoplasm. So this led us to the question, where do those processes, both DNA replication and packaging, occur in the bacterial cell? Are we getting a massive remodeling of the cell? What's going on? So it's not unexpected to see. So this is a happy cell. It has got two nucleoids. So the experiment here is that we made thin sectioning, treat the thin sections with DNAs, and we see where the DNA is because it was digested. So within 15 minutes of infection, this comes from here to this situation. So we are looking to total DNA. So I don't think it's a predator, but it's a mess. Anyway, and this is supported by just TAPI staining to make it more colorful. So what we found is that the bacterial nucleoids of the cell, they lose their special localization, and during the process, we don't know exactly what happens. We look also by labeling where the bacterial DNA is. It's kind of pushed into the corners of this big compartment where DNA is, but it's still there. But we also know that it gets destructured. So normally there is a specific folding of these bacterial nucleoids, and it's lost. And we may show that by a variety of different techniques. And of course, we can see that there is a huge area occupied by DNA in the infected bacteria, which correlates with a large amount of DNA synthesized. So now to see where this sp1 DNA is done, this is a typical experiment to label specific DNA in cells. We just build a recombinant phage with LACO repeats, and then you infect cells that produce the LAC-IM sharing, and this shows you very clearly where the viral DNA is when you do live fluorescence imaging. We always try to have mono-infected cells, so a low MOE to make sure we don't have a lot of cells that are multi-infected, and you can see the non-infected very clear here. So we define this as the DNA compartment that is generated by viral infection. So the next question we want to address is, where do the sp1 DNA replication occur? It's surely there, but we wanted to know a bit more about localization and where packaging takes place. The underlying question is that when we have this type of compartment, we are going to subcompartmentalize these processes, or it's just spread all around? So to address this, and this is just an example of a long experiment that we made, is that we just had all the host replisome proteins labeled, the bacterial proteins fused to fluorescent proteins, and we also constructed fusion proteins to the to some of the phage replication proteins. And I will summarize a bit later what comes from the phage in the replisome and from the bacterium. And the experiment is that when you see in this example the DNA compartment, you can see that this is rather homogeneous, where you can label the specific with the phage DNA, but you get a punctiform distribution here of both of the DNA X protein, which is part of a complex that loads the clamp to the DNA for replication. And the same spotty organization is found for the S-phage helicase. So this is interpreted as we are having SPP1 host hybrid replisome synthesizing DNA. And this is not homogeneous in the cell, but you can get it spotty in some areas there. Of course, we are not seeing individual replisomes with the resolution we have in these experiments, but still it's not homogeneous. And if we base ourselves on the type of qPCR data we had got, and this is still the level of computing I'm able to make, you can see that you synthesize around 12 megabase pairs in 15 minutes, which means that you need to get around 13.1 kilobase pairs per second to be synthesized. Sorry, there is a bug here. This should be 13. I made a mistake. So, but when we look to the rate of synthesis normally of DNA in E. coli, which is around 1,000 base pairs per second, this shows that must be several replisomes acting in parallel to get synthesized concatenators in the cell. So if we now summarize all the studies, and this comes also together with some biochemical evidence from the Juan Alonso's lab and Silvia Iora, the SPP1 is basically relying in the bacterial replisome to replicate DNA. So after the specific protein of the phage recognizes the origin of replication, there is an helicase loader, that being GP40, against a viral protein, and this GP40, when it's loaded into the viral replication origin, will recruit replisome proteins by interactions both with the primase, DNA G, and also with one of the subunits of the clamp loader system, that is here DNA X. So in practice, what we have seen, and this is summarizing a lot of work that was made just with all these proteins that are identified here, is that essential and also not essential proteins of the host replisome are recruited into this foci. So they take it out of the bacterial DNA, and it goes into this replication foci inside the viral DNA compartment. This is recruited by these protein-protein interactions. This was reported from biochemical studies. And we conclude that this viral compartment has a series of parallelized DNA synthesis factors that are in discrete positions of the DNA compartment. So now that we have been synthesizing the necessary concatenators to synthesize to be packaged, the question is now where packaging is occurring. So for that, and I think for this audience this is probably more interesting, we use an approach that was to label, to make fluorescent proteins, to components of the viral particles that are present at specific stage of assembly. So that we made fusions to the scaffolding protein of SPP1 of fluorescent proteins, and this allows us to specifically visualize procapsids. It's not always the case. If you want questions, we had some technical interesting issues here. And we also use the labeling of protein that decorates the capsid outside. And this protein, the sites for their fixation, depends absolutely on DNA packaging. So we have a real fluorescent marker to track where the DNA, where the viral particles that package DNA are. So we use these tools, and we start asking the first question, where procapsids are. And this is an image that I already showed to you. So this is an immunolabeling experiment with anti-capsid antibodies, but this cell is infected with a mutant that is blocked in DNA packaging, so we can see only procapsids here. And of course, you'd expect to see really the structure. The problem is that whenever we do thin sectioning and any embedding approach, we don't find any more the viral particles. We have no idea, so I think we need, this is definitely a good object for cryo-FIB-SEM, which are now implementing, but what we see is only the labeling and not the real particles, and this is a technical issue. And after validating this distribution of the procapsids around the DNA and also in the DNA area, but they are enriched at the periphery. And this is also shown here where we see in this case, the scaffolding protein as a proxy for fused to M-citrine for the procapsid localization. You can see they tend to be at the periphery of this DNA compartment. So when we look to the whole thing together, we are getting replication in different areas of the viral DNA compartment, and procapsids are localized here that we interpret as where DNA packaging might occur. Also, it's not a proof, but that's the best we can do at the moment. So the last question we had is what happens after DNA packaging? So for this, we take advantage of this GP12M citrine protein that we can code both from the phage itself, from the locus of this gene 12, or from the host bacterium, and ask the question where this decoration protein is localized. Again, the same approach. This is showing that we also make control, so we blocked DNA packaging. You can see here the replication, the viral DNA compartment. It's rather homogeneous, the distribution of the GP12M citrine, that this is now free because the capsids did not mature yet to the level that the protein binds. And in the overlay, you see just what we would expect, green through everywhere in the viral DNA compartment. So, but now when we make the full infection cycle, things change. You can see that you get again the viral DNA compartment, but you now get a very strong signals in defined areas that correspond to what we define as the procapsids. Okay, got the message. So in this case, you really have this strong signal from the decoration protein that we interpret to be viral warehouses where all these viral particles go. So you package in this viral DNA compartment, but then particles are segregated into the outside area of the cytoplasm, and they form again another type of compartment there. And we visualize also this by electron microscopy. So these warehouses localize on either side of the viral compartment, not much overlapping to it. So to put all this in the context of the full infection cycle, what we made was to look through the complete infection process in a microfluidic cell. So we immobilize bacteria and fluid medium through it to maintain the bacteria stably and metabolically active. We injected phages, and unfortunately we could not control them out, so cells get multi-infected. But you can see the process, the temporal process, that you get all these DNA replication compartments that appear, then these warehouses that start appearing at around 32 minutes, things grow, and then lysis occur. You can see that these are ghosts, so some of the structures, essentially the viral DNA compartments, start disappearing, and then you get lysis. Interestingly, and when we look to these images, cells still continue to divide and they continue to elongate. So they still have the capacity to continue to live their life while they are being infected, but the final result is always the same. And here we have a long period because this was done at room temperature experiment. So to summarize what we observed, this is essentially the description of how the phage is compartmentalizing the cell during infection. So it doubles the total amount of DNA in the cell. This goes in a defined focus in the cell, which would be expected. I mean, all these different concatenators get together in a defined position. And then later during the DNA, during the cycle process, you accumulate procapsids in the periphery of the cell. If you have blocked DNA packaging, they stay there. If then they package DNA, they are segregated to build these DNA-filled particles warehouses. So this is just a view what's going on. And what we are now pursuing is to try to understand how it starts happening by make single particle tracking of the entering DNA molecules before it is established in a defined position in the cell, to follow the DNA compartments to know if this is phase separation, some type of polymer collapsing that is attributed for DNA, or any type of aggregation for the procapsids. So that's the type of questions we are asking. And finally, we want to see the individual replisomes, and for that we'll need super resolution approaches to see both distribution and dynamics of these single complexes. And just to finish the talk, so the people that made the work, so Lina Yakutit started during her PhD thesis this research. She made the initial construct. Audrey Labart picked the project and this made, for her PhD, made all the constructions and optimized all the imaging to make this stuff. Ruth Carvalho Lopez, she's a specialist in B-settlist cell biology and high-resolution imaging approach. Sylvia, she's the expert in DNA replication. Torsten Milk and Beatrice Fowler, Torsten. Rudy, if you remember from the old times, they are in Berlin. They made the thin sectioning work with Audrey, and finally Rick is the specialist in qPCR. And this is our team that during part of the time that this was done. And I think Stefan will present tomorrow. You will see him without mask, and he will come to the fundamentals of structural biology and so on. And I thank you for your attention.
Thank you for that really interesting talk. We do have time for a couple of questions. Yes, at the front.
So do you have an idea of what keeps the warehouse together? Is there some kind of cytoskeletal component involved?
No, it's most probably aggregation. In fact, we can reproduce this in vitro by just getting these structures and putting them together with micromolecular counting agents. So we think this is really a matter of these particles tend to just get together. And when we have particles with and without GP12, the behavior is different, telling you it's really a surface properties issue.
Question over
there. Nice talk. I would bet that the replication is a fluid compartment, but you'll see. But the area, the virions, the sort of storage of the products is like parking lot for virions in the factory. But probably is semi-crystalline arrays. We see that in virus, for example, it's been seen for rotavirus as well. Close to these factories is this sort of parking lot. Do you see any of these in sections? Because that's really like it's sticking out.
We don't see it in sections because we have this problem when you do sections, you need even if we do the stock Waiazu procedures that you freeze stuff without fixing before, then you have a post-fixation step. And then this destroys the integrity of this. You can see them in cryofipsem, but I didn't have these results. So you can really see these, like it was described in the old times for herpes virus, you get these nice networks of capsids getting together.
Okay, so can we thank Paulo and all of our speakers again? Thank you all for keeping to time so well. So now we have lunch, which will be in the same place as yesterday, and the program will resume at 12:30.